everyone. Sorry, my brain just a bit swollen from all the information that Take just gave. Amazing talk, Take, making me feel very underprepared. Um, anyway, so my name is Bea. I'm a research technician at CSIRO. I'm based in Canberra, Australia, but I'm originally from Thailand. So can I get a little bit of reaction from fellow Southeast Asian? I just want to wake everybody up just in case. Thank you. Thank you very much. Oh, thanks. Okay. I tried to. Oh, for some reason. Oh, there you go. So just a bit of outline of my talk today. So originally I was going to give you a bit of introduction of the FAW armyworm, but Dr. Tekte just did amazing job on that. So we're just gonna skip that. And then I'm going to talk about a little bit of the traditional way of identify biocontrol candidate for any pest. And then I will jump to kind of how do we use genomic to help us understand a bit more about biocontrol agent that we're working with, and how do you use the genomic to identify potential bioactive compounds. Just to let you know that this talk is kind of a bit of showcase and give you a bit of pipeline. How do you do that? Rather than I'm trying to present my work. So I don't want you to get caught up too much about my data, result, or detail, but a bit more of general idea. And then at the end, conclusion and question. So just let you know the example that I'm going to use today. It was the data from the joint project between Macquarie University and CSIRO that I led. So the project was kind of formed with the idea that we try to identify endemic, like Australian or New Zealand biocontrol agents of for the fall armyworm. And the candidate that I tested is entomopathogenic fungi and Bacillus thuringiensis or BT. So the very easy or very traditional way of identify any biocontrol. First step, bioassay. So you got your fungi, you got your, pest, caterpillar, or whatever pest you're working with. Check and see if it's work. Then you'll be able to find, isolate that work very well, isolate that work not so well. You get your number, you isolate that quite effective. Similar with the BT, you have your BT, you do your bioassay, you get your result. But then what next? You narrowed out the isolate the strain that you would like to go further, but what you're going to do that afterward. This is kind of highlight all the purpose of my talk today. I just want to show you how did I use the genomic to help identify biocontrol candidates and also potential bioactive compounds or, in BT case, BT toxin. And this subject will be divided into pretty much two sections. So first, identify bioactive compound in fungi using a piece of software called anti-smash. And then I will give you a bit of example, how did I use the genomic to identify BT toxin sequences. Anti-smash. Have anybody used or heard of anti-smash before? Can I get a bit of reaction? Just in case. I just want to see like what sort of the audience. Okay, very quiet. Nope. Okay, all right. So anti-smash is kind of bit of software that you can run through your local high performance computer or HPC. But if you don't, oh, I got a little bit of thumbs up. Awesome. But if you don't have access to high performance computer, that's okay. You can also use through the website, which it look like that. So I really encourage people to, go to Google, search anti-smash and play around with that. So the question is, what do you need to do that? So for the fungi, if you would like to try to identify, potential bioactive compound through that genome, you need your genome and you need annotated genome, which is FASTA file and GFF files. And for the bacteria, you only need a genome. But if you don't have the genome yet, like for example, you're quite new to the genomic field, that's okay. I highly encourage you to go to NCBI website, select genome, type species of your interest or genera of your interest, download the genome. For the fungi, you will need FASTA file and GLF file, and for the bacteria, you just get FASTA file. And then go to anti-smash website, upload the data, and click submit. It's easy as that. It's very easy and straightforward. What do you get from that? So this is the result from anti-smash run. So you get pretty much biosynthesis gene cluster of your whole genome. The first row, actually, can I get a bit of maybe a little bit? No. Okay, I'll just try to get a little bit of cursor. Can you see my cursor here? Okay. So you get a bit of region and the type of secondary metabolized. Then you get literally the location of those cluster. And is that those cluster match to any non-cluster that previously identified? And then how similar to the cluster that known? So you can see this is, I think, is a Metrorhizium fungi. And you can see there's a lot of known cluster, but also a lot of unknown cluster. And if you're working with something that, under research, you probably kind of expect to see not many known cluster. Having said that, again, it's a very piece of software that very easy to use. And if you're interested, please give it a go. And when you get the result like this, I also highly encourage you to click and see what it look like, because sometimes it can give you maybe 95% similarity, but when you click to it, there might be like promoter missing in your genome or core biosynthesis gene missing, which really assume that that will not gonna work, or even though they have a low similarity, but core gene are quite similar, it might work. And this is just one of the so sorry, this is kind of example of my results. So this is a metrorhizium isolate that I tested again for armyworm. And for the bioassay results, we found that M0121 worked exceptionally well compared to other metrorhizium species. And when we look at the genome through the anti-smash result, we found that there are one very interesting cluster called dextrosin A, which we know that is have some sort of insecticide property. It doesn't mean this is the reason why this particular isolate work better, but it could be. And this is kind of a starting point of further research to confirm this hypothesis. It could be, LCMS, transcriptomic or proteomic or many other research to follow up. Moreover, we also found that there are one very interesting cluster that pretty much found in all of the metrorhizium isolate that I test, and that one called Swansonite. Swansonite is potential pharmaceutical drug for treating cancer. So first minute you might working on biocontrol, and the next second you might be working on cancer research. I'm just kidding. But what I'm saying, and I want to show you that using the piece of software like anti-smash and genome can really help understand and see the potential of your isolate. Another interesting piece of information that I found is this particular metabolites, also known to poison goats, horses, and cattle in many places, which are very bad news because if you're going to develop any biocontrol agents, you don't want them to produce anything that could be harmful against, mammal or other species. It doesn't have some cluster in the genome doesn't always mean that they will produce that particular metabolite. but it also tell you that, we got to keep eye on that, or, maybe we have to knock out this particular gene to make sure that it's not going to produce something nasty in the wild. And next is, how do I identify Bt toxin sequence from the genome? This is a little bit more complicated. For some people here might think this is still very easy, but I just don't want to go too much into the technical detail. I'm just gonna give you a bit of pipeline how I did that, and if anybody really interested in doing the similar thing or would like to have a chat, feel free to email me or Dr. Tech or any of us, and I'm sure we can help you go through, doing that. So to identify the Bt toxin sequence, first we need a genome, and we need annotated genome. When you annotate genome, you will get pretty much just sort of the gene names of particular gene that you have. What I did is pretty much just extract the sequence that was kind of annotated as cry protein, crystal, VIP, or protein that we know that might be toxic against Lepidopteran species. And then I confirm the results by blasting them against GenBank. Annotation most of the time are not perfect, and it's it's quite common that you need to follow up to make sure that that is the right annotation. And when, especially in BT field, when you go to the gene bank, you'll find that it's very messy. One of the gene that from the, my BT that I tested is called BT05 and it's sequence 3. So I named them BT053. Matched so many cry toxin. It could be cry1H, cry1G, or cry1D. And how do I identify which actually is? So I use phylogenetics to help confirm what toxin they are. And from the phylogenetic tree, I found out that BT053 is actually cry1H, and we still couldn't identify whether it's cry1HA or cry1HB. And this is pretty much the result of the BT that, like, pretty much successful candidate against fall armyworm. So unfortunately, we found that most of BT that work against fall armyworm contained toxin that we already know that is sort of toxic against fall armyworm, or fall armyworm susceptible to those toxins. So it's not really that exciting, except one of the BT. So this BT called BT02, we found that there are five sequences that will sort of have the structure that look like cryotoxin, but it doesn't matches any known toxins from GenBank, which it kind of give us a little bit of idea that, okay, might be a new toxin that also work against fall armyworm. This is really exciting new and obviously require further, follow up, maybe could be expressed in a different microbe like E. coli and test again for armyworm, like, using pure protein to make sure that this the new toxin that work against for armyworm. Pretty much that's it. Very short presentation. So to conclude this, I just want to show you that Genomic is actually very powerful tool to identify potential insecticide or Bt toxin, or generally speaking, to help we understand a bit more about the species or, biocontrol that we're working with. It also helps us identify potential harmful chemicals and keep us, help us keep it in mind before further develop that biocontrol agents to actually, use in the field. And this is just a few of many examples of how genomic can be used. That's it.
Thanks, Bea. That was fantastic. Really nicely presented as well. So well
Thank
done.
you.
You've got lots of feedback there. I see on the screen from your fellow colleagues across the region. Just a quick question because we're sort of a bit tight on time, but we'll come back. So what are your next steps now? Because you've finished your research here, and so how are you taking that forward? Just sort of quickly, what are you looking at now?
So this project is pretty much finished. I think let's talk about that with Dr. Tek Tay.
But following on
Okay.
from what your findings are, what do you think would be the key things to look at now? Because you've found some really interesting and exciting kind of things to focus on next. So what do you think is the next step?
For the BT, I think the very next step would be probably express in E. coli, and we could try the pure protein against, for armyworm or different insect species, because we don't really know what's going on. I did actually try to find if there are any outer metabolites that have insecticide property in that particular BT isolate, and we didn't really find anything either. Having said that, it's a lot unknown in the genome, so it could be that one of the five work really well or work together, or totally something that we really don't know about. Yeah.
Yeah. Tet, did you want to add to that? You're on mute.
You think that we'll learn after COVID? No, no. Beatrice has done a really fantastic job. She was a delight to supervise her PhD, and has provided examples of what with a bit of support can be achieved. I think across respective countries in all the audience, I think there will be a lot of opportunities to develop unique and localized or regional relevant solutions for biological controls, and support for genomics look towards Beatrice, for example.
Excellent, great, thank you. And it's a good reminder just of how we can use genomics as well. And we'll come back to that in the panel discussion. Thanks, Bea. That was a fantastic presentation, and I put a link to the report today as well for everyone to look at around your work. So thank you very much.